Hello and welcome to another bonus video. This video will be dedicated to the question of what are RILs. This will be a second part to the bonus video saga that will be a lead up to a major video that I'm working on. Let's get started. What you're seeing is the base form of a self-pollinating crop breeding project. Basically you crossbreed two varieties to get the F1 generation. It's from this generation that all the seeds will be collected. The next generation will produce the most variety out of all generations. This is because, although there's no more cross-pollination, the process of meiosis, which is the genetic reshuffling process that happens to create sperm and egg cells, still creates enough genetic variation that even though the self-pollination process happens, there's still some variation happening due to the recombination effects of sperm and egg, or in this case pollen and egg, within the plant itself. It is during this time or at a later date when individual plant lines are discovered and mass propagated until they reproduce true to type. When you mass propagate a specific phenotype and only select for a specific individual phenotype, eventually that genotype becomes fixed into the gene pool as an individual genetic line, due to the recombination events becoming more and more rare with each subsequent generation, as meiosis without outside genetics reaches its limit. This process usually takes 12 generations to reach 99% genetic homogeneity. By the 7th year, it's worth testing as a variety as reached around 95% genetic homogeneity, with only small amounts of off-types showing up within the population. Within the real population, like what you see right here, the goal is to homogenize as many varieties or genetic lines as possible to maximize the amount of variation you have access to. Now, why would you do that? In genetic experiments, like what we talked about in the previous video, you need a lot of phenotypic data in order to associate a specific phenotype to a specific genetic region, especially for a quantitative trait. Indeed, you need at minimum 100 genetically distinct lines derived from initial cross to get the resolution you need to find a connection between a specific phenotype or physical character trait and its genetic package. It is through this process that experimental lines used for breeding stock and specific genetic regions are discovered within a self-pollinating crop species. Indeed, you can see a great amount of variation just within this population itself, even though these are all sibling lines. This is within the growth chamber, where all environmental and soil character traits are controlled to the utmost degree. So all the variation you see is mostly due to the genetics themselves, not because of any inherent aspect of the environment. Stay tuned in the future for the big video that will likely last about 20 minutes, and a further follow-up video that will likely be exactly an hour or more. Until then, thank you for watching.